our last speaker. We kept him till the end because we know that everybody will wait for his lecture. So he doesn't need introduction, and we're very glad to hear his thoughts about the future. When we asked him about the future of pain medicine, he added far future of pain medicine. So he's beyond the future. Please, Dr. Marshall.
So beyond the future. When Prof and Devor asked me to give this last talk in the future, I knew this was going to be a punishment. We're very poor at predicting the future, and the farther the future, the harder it gets. Who would have thought that, or predicted, I don't think anybody, that we would all be walking around with a powerful computer in our pockets, or that we could come to the middle of the Arabian desert and be in a place like Dubai. And it's actually worse than that. An important statement, the Gemara, says that after the destruction of the temple in Jerusalem, the power of prophecy means predicting the future, was given to drunkards. That's people who drink too much wine. And that's the way I feel. But when a guy like this, handsome, silver-haired, asks that I do it, what choice do I have? So here we are. And I begin with a premise. The premise that general anesthetics permit pain-free surgery. That is, you can do any kind of noxious stimulus, and the individual under general anesthesia will not feel it. So general anesthetics permit pain-free surgery, albeit with side effects. We can discuss the side effects, of course, sedation and whatever. And on those grounds, they're the strongest, these molecules are the strongest pain relievers of all. And that brings me to my prediction of the future. So I'm going to guess that in the future, we will be able to understand how anesthetic molecules actually accomplish this, how they eliminate pain while preserving life-sustaining housekeeping functions. And the guess is that when we understand how these molecules permit pain-free surgery, that this will lead us to the ideal pain reliever, free of side effects, a perfect morphine. So let's see if I can convince you of that, Rob. Our story begins 170 years ago with the initial demonstration of general anesthetics at the Ether Dome in Mass General Hospital in Boston. And as we all know, many of you, most of you, I suppose, are anesthesiologists, there are four main effects of anesthetics, of general anesthetics of all sorts. Motor suppression, LORR, means loss of writing reflex, usually an indicator in animal experiments. Amnesia, loss of consciousness, whatever that means, and loss of pain sensation. And I'm actually going to be focusing on this connection, on the connection between the loss of consciousness and the loss of pain sensation, where I think these things are intimately connected, and we don't think often enough about how intimately these two things are connected. The earliest mechanism proposed for how general anesthetics eliminate consciousness and with it pain are that they suppress brain function in a general fashion everywhere in the brain to a level that the brain can no longer carry out the very high computational processes required in perceiving things, in being awake and being conscious. I have to stress a distinction which you all know about, but it really is central, and I have to start with it. And that's the distinction between pain and nociception. So nociception is a word that was invented by Charles Sherrington a long time ago, which refers to the response of the nervous system, particularly the central nervous system, to noxious, potentially pain or tissue damaging stimuli. So it's a response of neurons, the behavior of neurons. We talked about the pain matrix yesterday. It's a response of the nervous system to a particular strong stimulus, be it mechanical or thermal or chemical. Pain, in contrast, is an unpleasant sensory and emotional experience, and I underline the word experience. And the real difference between these is that an experience, that is pain, is dependent on the presence of a conscious brain. Pain is something that only occurs when there is consciousness. This distinction really should be present in all sensory systems, vision, for example. So if I approach a grocery store, there's usually a sensor there that detects either whether there's movement, or it could, in principle, detect a face or a body, and it opens the door for me. But this system is not seeing in the sense of visual perception. It's getting a visual signal, and it's operating a motor. And there's no conscious, no perceptual event happening here at all. And this word consciousness is really the elephant in the room, including in this room. We're talking about pain, but we haven't actually mentioned the fact that we have to be conscious to be perceiving pain, to feeling pain. And of course, general anesthetics wipe this out. So what is consciousness? So over the generations, going way back, we've had lots and lots of different definitions. Starting from the proposal that it's people on a cognitive stage, or it's a flashlight, spotlight. One didn't talk about this phenomenon in biological circles until quite recently. It's become, well, more in a mode. Today, certainly the most prominent theory comes from Thani, who says that it's an emergent property, that it's something that happens, that just comes into being if we have enough elements, enough neurons, and they're connected enough in a functional manner. So if we have these conditions, then consciousness happens. From my point of view, none of these are definitions at all. They're all metaphors. They're saying light. It's like a stage. It's like a beam of light. What we're really talking about is how neural activity, the activity of neurons in the brain, brings about conscious experience. It's something that the philosopher David Chalmers calls the hard problem. But we don't even know in what sense is it hard. Is this a hard problem, understanding this phenomenon of consciousness, because of its intrinsic complexity, like cracking the genome where there are a billion parts that you have to work out? Or is it something that just requires an insight? For example, the Darwinian theory of evolution. It's something you can teach to a high school kid in an hour or so, and he'll basically understand the principles. And it explains so much with this one idea. And I hope sometime that somebody may wake up in the middle of the night and say, God, it's such easy. Why didn't I think of it before? And if this happens to you, Grace, if it happens to you tonight, and you wake up and you realize what consciousness really is and how the brain does it, send me an SMS, please. I'd love to know. So defining consciousness is going to require understanding the algorithm, understanding how this computer up here actually does it. And I think that at this stage we have absolutely no idea, no guesses, no theories that merit the name that can account for it. And even if this guy were to come down from, it wouldn't be Mars, we know there's nobody living on Mars, but some other planet, and give us a disk and say, hey, put this disk into your computer, and the PC will become conscious. The computer might actually say, hi, I'm here and I feel pain. But we would have no way of testing whether it actually does, unless we actually could understand the mechanism, and that's what we have to do. On the other hand, easy to tell. With some exceptions, it's pretty easy to tell whether somebody is conscious or not, including an animal. So here we've got a picture of 20-odd people, and I bet everybody in the room will agree that there's only one who's clearly unconscious. It's that guy right there. So there are behavioral correlates of being conscious or being unconscious. There are also neural correlates. The best one is probably the oldest one, simply the electroencephalogram, where the patterns of someone who's unconscious, for whatever reasons, is usually quite different from the cortical patterns of someone who's awake. Although here too there are some exceptions. So putting aside definitions and algorithms, we all experience consciousness. I hope that all of us are conscious right now. It's the me inside. That's what consciousness is. I don't think it even has much to do with being conscious of particular things, although, of course, we are normally. So when I look out at you, I would have said 50, 60, most of my attention or my visual, my sensory input is visual, and there's some sound and some sensory and proprioceptive, of course. But I don't think those are critical to the phenomenon, the basic fundamental phenomenon of consciousness, which is the thing that is required for pain sensation. So what do I mean by that? So here's a famous picture from 100 years ago by Ernst Mach. He's drawn a picture of what it looks like when he's looking through his eyes. And we are all actually looking through our eyes right now. And you can even see up on top, he's got an eyebrow and a mustache down below. That I guess you can't see through your eyes. And he said that the he inside, the me inside, is somewhere between your eyes and maybe 2 or 3 centimeters back. So you can actually locate where the me is. And I feel the same way. I think that's where I am. Now, I'd like you all to play along with this and think about where the you inside is. So please, together with me, just close your eyes like that. Your eyes are closed, so probably 60%, 70% of your sensory input is gone. Are you still there? I'm talking to you right now. Are you still there? I'm sure you all agree you are still there. And if you were to cover your ears as well, you would still be there. You wouldn't be 70% less. You're all there, even though all the sensory input is cut off. So when we normally look through our eyes, we're seeing a scene. And my little computer in my pocket. So here it is. And it's actually seeing the same thing I'm seeing. But I'm perceiving you. And this little computer is not perceiving at all because nobody put perception into its program. Now, what's all this got to do with predicting the future? There are brain state transitions which completely eliminate this perceptual thing, this thing that we call consciousness. Certainly falling asleep, fainting, for whatever reason. And pharmacologically, we can do it in a very controlled manner by delivering a certain dose of particular molecules. And what that does is it changes, and very rapidly, changes our brain state from perceiving, from seeing or feeling pain, to oblivion. We're not there, we might as well be dead. And with most of these phenomena, not dying of course, but the rest of them, it's reversible. We can come back and forth, and the back and forth can happen very, very quickly. I imagine you've all been at a boring lecture, and you know that you see somebody, or maybe yourself. So you fall asleep. You lose consciousness. The brain is turned off. You lose consciousness in a second, a fraction of a second. You lose muscle tone. That's part of the motor suppression, and your head falls, and that's enough of a stimulus to wake you up and bring you back. So you can turn consciousness off and bring it back on in a matter of hundreds of milliseconds or a second or so, something like that. And this phenomenon, the me inside, that can switch on and off is I think, the very basis of pain perception. And if we're going to get this perfect morphine, I think we have to learn, figure out how it works. So let's go back to the ideas about how general anesthetics actually work. I know this isn't much taught in medical school, even to anesthesiologists, but so you might find it interesting. So as I said at the beginning, the original idea is that we're talking about a generalized suppression of brain function, particularly cortical function. I like to call this the wet blanket hypothesis. There's a fire there, you just take a wet blanket and just sort of calm everything down all at once. Based on the Overton-Mayer correlation, which I won't go into, the presumption was that the molecules are actually acting on brain lipids, on membranes and membrane viscosity. And it took quite a long time before it became clear that that's probably not true, that the receptors, that the action of general anesthetic molecules is on proteins, particularly channels and receptors. So that was a change, that the target is not lipids but proteins. And very soon after that, convincing evidence from Joe Antonini that for loss of consciousness and loss of memory, the action of the molecules is presumably in the brain, and for loss of motor tone, because you really are flaccid, so the motor neurons in the ventral horn are suppressed. That the atonia and the loss of pain sensation is due to suppression at the level of the spinal cord. So we have a slightly changed icon for this. It's no longer generalized suppression, but patchwise suppression. Suppression in the brain for one thing, and the spinal cord for something else. But still, the presumption is that the molecules themselves have to get to the cortex or get to the spinal cord to do their job. Now, if we go back in history another step, in the research on sleep, rather than the research on anesthesia, in the research on sleep, a very different idea arose, and most of the credit is given to Maruzi and Magoon, particularly a paper published in 1949. That's a long time ago. And it was based on the observation that if the electroencephalogram is recorded, and this is in cats, if one removes all the sensory input from the body by cutting the spinal cord, the lower medulla, still the cortex oscillates, goes back and forth between a state of wakefulness and a state of sleep, of unconsciousness. However, if the section is made at the mesopontine level, just a little bit above the medulla, then the cortex is locked in a sleep, an unconscious state. And the conclusion was, or the idea, the theory was, that between these two cuts there's a region which actively drives the cortex into a state of wakefulness, and otherwise it's asleep or it's unconscious. And a similar pathway going down to the spinal cord, which controls atonia and pain sensation. Called the reticular activating system, this brainstem structure area. The icon I'm going to use is this, on the lower right. And the main difference between the previous icon is now the molecules, the actual anesthetic molecules, which get everywhere in the brain because they're in the circulatory system, but they only act in a localized place, according to Maruzi and Magoon, in the brainstem, or the mesopontine tegmentum, in the brainstem, and the rest of it is done by wires, by cables, by axonal pathways that go to the spinal cord on the one hand and the cortex on the other hand to change brain state and to allow pain perception, all perception, to occur. Most of the research on sleep mechanisms since then has been to detail the various subgroups of neurons within the brainstem that are responsible for brain state changes associated with sleep. And here's a modern representation of the circuitry. So we have a quite complex circuitry with lots of different nodes and pathways connecting these areas. Now, quite recently, about 10 years ago, not much more than that, the researchers who were interested, and there aren't very many of them interested in anesthetic mechanisms, sort of adopted this circuitry idea from sleep and said, and in fact this was said by Maruzi and Nagoon way back in the early 50s, that anesthetics, general anesthetics, might be acting on this sleep circuitry, actually hijacking the sleep circuitry in a way and causing a transition between wakefulness and loss of consciousness. So we have two very different theories here. One of generalized suppression and one of dedicated pathways. And here we are in 2023, and these are very different models, and we don't really know which may be operating. And this is where I'd like to report on some of the work we've been doing recently in our lab in Jerusalem on this subject. So the approach was to install in the brain of a rat a little cannula, a little tube, which allows us to micro-inject, to inject a very small amount of a general anesthetic. The original survey was done with a barbiturate and a barbital, but we've done this now with a number of other general anesthetics. And simply deposit a very small amount at a localized spot in the brain and ask the question, well, does this awake animal become unconscious? Does he become anesthetized by delivery of anesthetic agent to a specific spot? Now, since we didn't know whether there is such a spot or where to find it, and presumed that you had to do this bilaterally, because systemic dosing, of course, will affect both sides of the brain. So we simply did a survey, micro-injecting very, very small amounts of barbiturate bilaterally into the brain. Now, with this cannula, pair of cannulae in the brain, we can inject in that sort at these two spots, but of course we can go anywhere along the path of the cannula and take another rat and have the cannula separated a little bit more, and that way cover this whole part of the brain. So in fact, in a three-dimensional matrix, we covered the entire brain, looking for somewhere in which we might be able to get a transition between wakefulness and sleep or unconsciousness, anesthesia, with a very small localized delivery of the agent. And I'd like to show you the main result, hoping that this video will work. So here we have a rat. You can see on his head we've got the ability to inject, and we're recording EEG. At this point, Anne is now micro-injecting very, very small amount of barbiturate into the animal's brain, and within tens of seconds, the animal is out. He's not dead, he's very much alive, but he's anesthetized. And you can see the EEG has gone delta, and he remains that way for, well, 20 minutes, half an hour, something of that sort. We'll fast forward. The EEG now returns to a wake-like state, and a couple of minutes after that happens, the animal's back up on his feet and feeling fine. We found, in fact, only one spot, bilaterally symmetrical, in the brain where this happens. It doesn't have an anatomical name, so we simply named it by the location, the mesopontine tegmental anesthesia area. So it's on the border of the mesencephalon and the pons, and you can see where the blue dye that was injected, that's where it is. And then this is an injection of 200 nanoliters. A nanoliter is a very small amount. It's a millionth of an ml. So it's an amount that you almost need a microscope even to see it. We quickly discovered that you don't have to inject on both sides. It's enough to inject on one side to get this effect. And with a variant on the method I just showed you, which we call the bonus time method, I won't go into the details, it's published. We were able to get even better control of the injection sites. So we can now inject as few as 10 nanoliters, which really does require a microscope to see it. And what you're seeing now is a cross-section through the brainstem of this animal. And all of the red injections, which are very small injections, all of those gave the effect that I just showed you in the video. That is a rapid transition from wakefulness to consciousness and a reversal after some tens of minutes. And all the blue injections, which are some of them only half a millimeter away, are failures. That is, it didn't happen. And as I mentioned already, it doesn't happen anywhere else in the brain that we were able to find, except for this one little spot. We estimate that the small injections could not have exposed more than about 1,900, 2,000 neurons in the brain to this drug on one side. And I should point out that we're talking now about concentrations of pentobarbital which would be available in the brain, which would be in the brain after systemic dosing at a clinically relevant IV dose. You can immediately see that this is not compatible with the wet blanket hypothesis. Because if you had to spread the drug to the cortex and the spinal cord, you don't have nearly enough time to get there in the 20 or 30 seconds it takes for this transition of brain state to happen. And of course, the dose is far, far too low. If you inject the same dose IV, which of course we've done many times, you see no change at all. The dose is simply 1,000,000 times too low. The cells in this area look like the reticular formation. There's lots of neuropil, there's some big cells and little cells, and there's no secrets in just looking at the cells themselves. But we took this next step, which is called pharmacogenetics. We've actually heard this name mentioned today. It's really cool, so try to understand what we're doing here. So we now have located this spot, and what we do is we inject into that spot a virus, an engineered, a designed virus, which is able to cause cells that become infected in this area by the virus. The cells that become infected, they express a color, fluorescent or red fluorescent dye, so we can see which cells became infected. And in addition, they express a receptor. It's actually a muscarinic receptor, but it's a modified receptor that does not respond to acetylcholine in the brain or anything else in the brain. It only responds to a designer agonist, which normally has no effect in the brain. So we are able to excite, or using a slightly different virus, inhibit the activity of these neurons. So that's what the injection site looks like. And if we look a little bit closer up, we've got a bunch of cells that look like the cells I just showed you, but now these are cells that have become infected by the virus, and these are cells we can either excite them or inhibit them selectively by delivering this designer agonist, which we call CNO, which is called CNO. So the idea is to simply, after the infection has happened and the cells express the molecules they're supposed to express, you can see the red marker, of course. We simply inject CNO, we ask, well, what happens? Well, the most dramatic thing that happens is that if we inject an excitatory drug, that is, we're exciting these neurons that I'm showing you here, that causes about half an hour of general anesthesia. That is pro-anesthetic, general anesthetic. The animal loses consciousness, goes into delta EEG. Almost the same result if we inject a very selective GABA-A receptor agonist, or pentobarbital or propofol as well. If there's no drug there, there's none of this engineered receptor present, and we inject CNO, nothing happens. And the an interesting thing is that if we use the inhibitory drug, that is, we inhibit these red cells, nothing happens. So we've got a small population of neurons and we can see them, there's those cells, and we can measure them, and we can study them. If these cells are excited, inhibiting them doesn't help, and if we excite them, we get a transition to loss of consciousness, the ability to carry out pain-free surgery. Okay, so it's sufficient to excite these neurons, but are they necessary for getting general anesthesia when you dose systemically? I mean, is there anywhere else in the brain that this works? And this experiment is done by destroying cell-specific lesion of the MBT on both sides. Firstly, we expected this would cause coma. It does not cause coma. So this is not the proverbial center of consciousness where if the cells aren't there, the individual is not conscious. So we believe it's a switch that these cells are responsible for doing something elsewhere, probably in the cortex, because the EEG has changed, and probably also in the spinal cord, because the motor tone has changed. But they do cause a relative insensitivity to anesthetics, to the same general anesthetics, delivered systemically. And let me show you how that works. So here we're looking at the depth of anesthesia, where the top number is a deep surgical anesthesia, as a function of the dose that's delivered systemically, IV, to animals that are intact versus animals that have had the MBTA destroyed. And what you can see as we gradually raise the dose, when we get to about 40 milligrams per kilogram, which is the normal anesthetic dose of this drug in rats, you see 100% of the animals are anesthetic. This is not something that happens in some cases and doesn't happen in others. However, none of the animals with the MBTA destroyed become anesthetic. They all stay awake, and you have to almost double the dose, which takes us to close to a lethal dose for the barbiturate before the animal becomes sedated. So if you go high enough, you can still get an effect, but the animal is really no longer responsive to the clinically relevant concentration of the general anesthetic drug. This works for pentobarbital, which is the drug we used in the initial survey. It also works for etomidate and propofol, which are believed to act on the same receptor, on the GABA-A receptor. It does not work for ketamine and medetomidine and dexmedetomidine, which work on different receptors. So what we're concluding is that excitation of a small subset of neurons within the MBTA is sufficient to induce general anesthesia, and it's also necessary to induce general anesthesia at clinically relevant doses. And here's our working hypothesis about what's going on here. So GABAergic anesthetics, and I'm restricting it to GABAergic anesthetics. We simply haven't done research on the other types, which may involve a similar circuitry or may not. But GABAergic anesthetics, these given systemically, get everywhere because they're in the circulation, but they only act at a particular spot. Not unlike hormones, which circulate everywhere, but only act in certain tissues that have the appropriate receptors and connectivity. And in fact, the fact that you normally inject these drugs systemically, there are a number of side effects that we did not see with direct activation of these cells in the MBTA. For example, we didn't see temperature changes, and we didn't see changes in respiration in other of the autonomic responses, which I believe are probably side effects of the drug getting to other places rather than getting to places that switch the brain state back and forth. And secondly, we believe that these cells that we showed, those red cells that I showed you, are sensors of GABAergic agents, and they cause a switch, probably a flip-flop switch, because the transition can be so quick, as I mentioned earlier, a flip-flop switch between two stable brain states that are actually realized in other places in the brain, the cortex and the spinal cord, presumably. So graphically, there's our MBTA. Some of the cells there project down to the spinal cord and are responsible for the atonia of general anesthesia and for the pain control, pain suppression. And others project up, amnesia presumably having something to do with the hippocampal cortex, and consciousness presumably having something to do with neocortex. And we can ask whether these pathways are dissociable, whether we can actually see them. We are able to trace the projections of the connections of these cells and see where they go, using tracers that run anterograde from the cell body to the end of the axon, or that run from the end of the axon in the MBTA, and we can see where the cell bodies are that send those axons into the MBTA. So we can work out the actual wiring diagram involved. It kind of looks like that. So we've injected a tracer in the amputee, and you can see descending fibers quite clearly here and ascending fibers here. And here's a schematic of the projections of MPTA neurons. Several actually interesting targets in the diencephalon and basal forebrain, and other interesting targets going down. When I say the ascending targets are interesting, it's because they actually are known to relay to the cortex in this way. Now, let me just go back a second. Another thing that we know is that each individual neuron does not send an ascending and a descending projection. Each one either goes down or goes up. And in fact, if they go up, say to the basal forebrain, they only go to the basal forebrain, they don't go to the thalamus, and they don't go to the zona incerta. So each of those neurons has a very specific target where it sends its axons and releases its neurotransmitter and does its job. Now, that predicts that we might be able to have dissociation between the different elements of general anesthesia. And in fact, from clinical information, we know that this is probably very likely. For example, in narcolepsy, you'll get the descent, the REM atonia, you'll get the loss of muscle tone, and the individual will just fall, collapse, but remains conscious. So we're not getting all four of the things, we're losing one while preserving others. REM atonia upon waking, I've never experienced this, but apparently it's quite common where somebody will wake up and he's awake, but he's completely paralyzed for 10 or 20 or 30 seconds. A little bit scary, but apparently common, and it probably is just indicating that his cortex is woken up from sleep, but the descending atonia pathway is still functioning for some short time afterwards. And sleepwalking may be the most dramatic example, where you're unconscious and your EEG is delta, but you can get out of bed, go down to the refrigerator and make yourself a sandwich and then go back to bed, all unconscious. So the subunits, the elements of general anesthesia apparently can be dissociated. And here we're talking about the specific pathways that we suspect might be involved. So here we've got these cells, and we know that some of them project down and some of them project up. Is it possible that we could find the ones that are going down to the dorsal horn and presumably turning off pain sensation, and who knows, maybe even control those or activate those separately without activating the ones that go up and cause sedation and loss of memory? Maybe it's that those, or maybe these three cells there, those are the ones that allow pain-free surgery. And maybe we could activate them selectively. Now, is that even possible? Well, firstly, it's easy enough to find out which cells there are. In fact, we've done it. You just put a retrograde tracer in the spinal cord, it comes back to the MPTA, and some of these red cells will be marked by this second tracer. And the ones that are marked by the second tracer, those are effector nodes. Those are the ones that, when excited, do a job. And what job do? They do the descending one to the dorsal horn. Now, that would be a first step, and as I say, it's easy and more or less already done. The second thing, which is also doable, but we haven't done it yet, and probably I'll never do it, but somebody else might, we can study these particular cells, which are the ones that provide us with pain-free surgery, and ask, do they express some distinctive receptor, something that could allow us to activate them pharmacologically without activating the other ones? So we're pretty sure that GABA A receptors are the ones that activate all of these. But each of them are different, and neurons express hundreds of different molecules. And maybe there's some tag, some handle that we can get for the pain-relieving ones that we could approach selectively. And finally, if we had such a drug, and this is prediction of the future, and I'm not sure that I can predict the future any better than anybody else, but at least it's an idea that might, in fact, ultimately bring us to the perfect morphine that we're all very much hoping is possible. Why is all this interesting? Well, firstly, I think you might, I hope that some of you are convinced, at least within the limits of the fact that this is done in rodents, that of these two very different hypotheses about how general anesthetics work, I hope you'll buy that we have some evidence here that the dedicated pathways is probably the one that's actually working. That is, that Darwin gave us a circuitry in the brain to cause transitions between wakefulness and loss of consciousness, presumably in the interest of sleep, whatever that's good for. And these molecules that were discovered in 170 years ago, or were adopted 170 years ago, just co-opt the receptors that normally are activated by GABA in the brain, but now are activated by propofol in the brain. If this prediction is true, then we might be able to obtain surgical quality pain control without the other effects, without the sedation. Reversing that, there may be conditions of coma in which, if we were to modulate these neurons in a selective way, presumably in this case the ones that go up to the cortex, we may be able to bring people out of a coma if there hasn't been too much actual severe damage to the brain done. I suspect that the same circuitry which probably evolved for sleep is also part of simply of fainting. So we know that if you breathe too much CO2 or not enough oxygen, you'll pass out, you'll lose consciousness. But do any of you know actually how that happens? Now, what in the brain senses and how is conscious perception and muscle tone and the rest, how does that actually controlled? It's the same question, it's a related question to how general anesthetics work. And I don't think we know very much about that. I mean, I don't even know if anybody's studying it, or concussion for that matter. And for me, as a basic scientist, probably the most interesting implication of all this is that it may lead us into the circuitry of what consciousness is in the first place. That hard problem of the philosophers, that if we ask these questions using biology, I predict, I'm not a prophet, but I predict we may actually be able to solve these hard problems by tackling them biologically, because these phenomena are happening here, between our ears, right here in tissue that we study and are interested in. Some acknowledgements. General anesthesia was apparently used earlier than in Mass General in 1849, so we have to give credit to the powers above. I'd like to thank some very good students who've worked with me and collaborators from other places, and of course to thank you for your attention. Happy to take questions if there are any. We have a chairman here. You might need a mic.
Thank you so much. Thank you so much for the amazing presentation. I attended this amazing lecture in Turkey, Istanbul last year, but I had no time to ask for this question. This question is from American Board Review Anesthesia. Intraoperative awareness under general anesthesia can be eliminated by closely monitoring electroencephalogram, blood pressure, heart rate, bispectral index, none of the above. Do you feel none of the above is the correct answer? And this is because no one of this machine, especially PIS, can study this point, MTPA?
I guess the short answer is I don't know, but of course I'm not going to leave it at that.
The correct answer is none of the above. That's with the textbook.
I'm not sure the textbooks always have an answer or the correct answer, particularly on these subjects. These subjects are really research issues. There's no question that the electroencephalogram is a correlate. But the electroencephalogram is an extremely crude measure of brain activity. We're putting big electrodes on the hair there, not even where the brain cells are. I mean, it's a centimeter away from where the brain cells are. And each electrode is picking up the activity, the summed integrated activity of probably 1 billion cells that are all firing in their own way. I like to think of it as there's a pond and there's lots of fish in the pond, and what we're reading is the ripples on the surface of the pond. Can we really learn about all the different kinds of fish and how they're acting by the ripples? If all the fish are going either that way or that way or that way or that, going together, synchronized, then we'll get big waves. But anything other than that, we'll get some very complex pattern. And it may very well, it probably does correlate with being conscious or being unconscious, but not necessarily. So if cells in a critical place are doing something different, and the ones up here, which are less relevant, are firing together, there would be a dissociation between the EEG and conscious. And I think that sleepwalking may be an example. I don't know really. So if we're talking about clinical anesthesiology, then you do the best you can. And if you've underdosed, the person may be awake enough to even hear and understand, although he may not be able to speak or move or tell you, especially if he's paralyzed. So it's a practical problem for operating room anesthesiologists. We all know that. But I'm not sure that it tells us that what exactly the relation is between a particular sort of analysis of the EEG and the person's state of awareness. We're not there yet, but maybe we'll get there eventually.
Yes, last question from the Congress. Our friend from Spain.
No, excellent presentation.
Thank you very much.
I want only one consideration. What is your opinion with the use of the magnetic and electric transcranial stimulation? Because it's possible to use and to measure by tractographic the change before and after.
We're talking about transcranial magnetic stimulation.
Yeah, magnetic.
Well, certainly magnetic fields can induce electrical activity, and you can certainly activate neurons to a certain extent focally, and I'm sure that this will improve so that it will become better focally. I mean, at conferences you often see TMS machines. You can hold it up to your own head, and you can get a little bit of a twitch in the place. So I don't think that's surprising. I'm not sure if we're going to be able to get to the brainstem, and I think that's probably a good thing, because I'm not sure that we want to start manipulating activity of cells in the brainstem. Cortical injuries, and we're talking about a very reversible, momentary thing. It's not a lesion, but cortical lesions almost always leave the individual awake and hopefully speaking. You have to have a pretty big lesion before you get serious clinical effects. And you rarely get loss of consciousness from simple cortical lesions. The lesions that cause loss of consciousness tend to be brainstem lesions. And in fact, if you look at them summed up, and there are several papers which do this, what is common between lesions in humans now, which cause coma, they tend to more or less overlap with this MBTA area that I've just shown you. So I'm not sure I'd want to play with that on the brainstem, but who knows, for a brief moment it may just put you to sleep and you wake up again. The fact is that you can even pretty much stop all brain activity. Severe hypothermia, for example, or very high doses of barbiturates can pretty much quieten down almost everything in the brain. And if you keep the person alive and breathing or whatever, it'll wash out and he'll come back and be the same person and remember his grandmother. There are a lot of mysteries about the brain, and really interesting mysteries about the brain, how this all happens. And we have to be modest about what we actually know and where we're going. So, Rob, that's the future. Thank you very much.
Thank you very much. It's a great closure to our Congress. It was a great adventure. We made it after some time, after the COVID era. WSPC is glad to keep in touch with you. As a new president for the WSPC, I will make sure that this level of program and expertise and a high level of speakers will be always there. We'll see you next Congress. We don't know exactly when, probably 2025. But we will be in touch because there are some small meetings in the region here to enhance this knowledge about pain management in for our community. Thank you very much for attending. See you soon.